Hello and welcome. I am Shailesh Appukuttan and I will be taking you through this step-by-step walkthrough of the EBRAINS live paper platform. In this tutorial-cum-demonstration, I will be showing how to access a previously published live paper. All you need to follow this tutorial is an internet browser such as Chrome or Firefox, and for certain selected features, you would require an EBRAINS account. The same can be obtained via the URL mentioned here. The live paper platform provides free and open access to all the published live papers. Users do not require an EBRAINS account to access any of these pages. The Featured Live Papers section presents a subset of the published live papers that are demonstrative of the various functionalities that are available through live papers. If we scroll further down, we see the listing of all the published live papers ordered by the year of publication. Live papers can be searched based on their title, the authors, the publication year, and so on. Let us search for all the live papers involving CA1 pyramidal neurons. We find that there are 4 such live papers. Let us take a look at this one from 2018. Here, we see an individual live paper. The top part of the page presents basic info such as the authors, their affiliations, the person creating the live paper, and every live paper that's published receives a DOI, which is indicated here. Further down, you see more info about the corresponding journal publication and its abstract. Scrolling further down, we come to the core of the live papers, that is the scientific resources. For this particular live paper, we see that the authors have shared the morphological reconstructions, the electrophysiological recordings, some test simulations that can be run on the cloud, along with the actual source code of the models developed and employed in this study. Let us take a look at each of them more closely. Here, under morphologies, we see that the authors have provided a large listing of all the morphology files that were used in developing the models. For each entry, we see that we have the option of downloading the corresponding file and also exploring the 3D reconstruction live within the browser. Clicking on this link takes us to the integrated 3D morphology viewer tool, which allows us to examine and explore the 3D reconstruction in detail. The tool provides various features such as hiding or highlighting various parts of the neuron, as well as saving the morphology file into different formats. Coming back, let's take a look at the electrophysiological traces section. Here, we see that the resources are classified into different categories based on the cell type, for example, pyramidal cells and interneurons of different types. And each of these sections contains a large number of data files. By clicking on any of these files, we get access to the embedded Neural Activity Visualizer tool. This can handle a large number of file formats and allows users to view and explore the data within these files from here within the browser. As before, users can also download the files by clicking on the download button here. In this next section, the authors have provided a link to an external resource and also the ability to launch live simulations from within the live paper. Let us try one of these. Clicking on the button launches Neuron as a Service, a web application that allows us to simulate single neuron models from within the web browser. This tool also provides a vast number of features. For starters, it allows us to view the 3D morphology of the neuron like this, or also as a dendrogram. Further, it allows us to place a current injection stimulus at a particular point on the neuron, then set the parameters for the stimulus such as the delay, the duration, and the amplitude. We can also specify the locations on the neuron from where we wish to record the membrane potential during the course of the simulation. Finally, when we launch the simulation, we see the voltage versus time recording for the various regions that we had specified. Users are able to download this data for further analysis. In this final section of the live paper, the authors have provided access to the source code of the various models developed as part of this study. The download button allows us to download the source code for each model. Additionally, the view button provides access to dedicated model catalog pages for each of these models. These pages provide additional metadata about these models as well as access to validation results associated with these models. Let us quickly jump to another live paper to see how Jupyter notebooks can be shared through live papers. This live paper provides access to 3 Jupyter notebooks. 3 actions are possible here. We can simply download the Jupyter notebook to have it run locally. The other 2 options require us to have an EBRAINS account in order to launch the Jupyter notebooks from within the EBRAINS Jupyter lab. The difference between view and clone is that the former opens the original Jupyter notebooks shared by the authors, where you can view the contents but cannot save changes to them. When we click on clone, the resultant pop-up indicates that we require an EBRAINS account and that we have access to a collab. Later in this tutorial, we shall discuss about creating an EBRAINS account and give you a quick overview of the EBRAINS collaboratory. Clicking on Proceed takes us to another integrated tool which helps us clone this Jupyter notebooks into our own collab. Here, we first specify the target collab and the path in that collab and click Continue. And finally, by clicking on Clone File, we can copy over these Jupyter notebooks to our own workspace where we can make changes and run them. Before we finish, we quickly take a look at another live paper just to demonstrate that live papers can accommodate various kinds of data. As part of this live paper, the authors have provided interactive 3D visualization of protein structures and also provided tabulated data associated with the published study. With that, you now have a good idea about what live papers are and how useful they can be. That is the end of this tutorial, in which I have demonstrated how to view published live papers and how to access and explore the resources provided in these live papers. So go on to the live paper platform and check out some of the existing live papers. EBRAINS live papers have been developed through the efforts of many different people with financial support from several organizations. I would also like to thank the developers of the integrated tools and services. These have been mentioned on the documentation page. For more information, check out the documentation at livepapers.brainsimulation.eu/docs. If you have any questions, you can contact us through our GitHub repository, eBrains support, or the eBrains community.